But it was writing.
Well, it's a real pleasure. It's a real pleasure to have Dr. Shivkumar Kalyanam with us today. And Shiv, as he's called by some, comes to us from Los Angeles, from this fancy Geffen School of Medicine, Geffen School of Medicine, UCLA, where he is the director of the interventional cardiac electrophysiology and a few other things. And Shiv received his MD from the University of Madras in India, and then after that continued some period of training in India, came to this country. I remember from the CV that he spent some time in Henry Ford Hospital in Detroit, then UCLA, and climbed up to different positions there with an interlude in the University of Iowa. And finally settled down in UCLA, and that's where he's coming from. And he'll tell us about, he's done a lot of work in clinical research associated with ablation and many aspects of interventional cardiac electrophysiology. He also got a Ph.D. on the way, by the way, that I forgot to say, with also UCLA. So, in physiology. So, without further introduction, the topic will be catheter ablation of VT and beyond. And it's the beyond that I'm curious about.
Jerome, thank you so much for that kind introduction. It's been a fantastic day to be here to see your work and FMOB, and of course Dr. Damiano, who I've known for many years, and because the EP program at WashU is, the most cited program really, because in terms of science, it's a program that has changed the entire approach to cardiac arrhythmias. Starting in 1914 with critical mass hypothesis to a surgical procedure for atrial fibrillation. This is a rare honor. Thank you for the invitation.
That was before my time.
So we know where the bar is for you. You're up there. So at least you have to come up with another hypothesis for fibrillation. But anyway, it's an honor to be here. I've intentionally chosen a topic which hopefully bridges both worlds. At the very end of this, we'll talk a lot about the neuromodulation part of it, but I'm not going to bore you with exquisite details of the most recent experiment, but I'm trying to put this in perspective for this group. So we have to thank, of course, NIH, the ever-shrinking source of funding for us, but much sought after, which is the only way we're able to run our programs. I'll talk a few words about clinical approach. We don't have to waste time on that. Talk more about substrate, structural components, and of course, autonomic regulation, which has been the big passion of our own research group. And again, it's a field where we're going back to the OR. We're going back to work with the surgeons to take the field to the next level. So VT can occur in the absence of structural heart disease, or it can be seen in the presence of structural heart disease. The type of VTs that you see in the absence of structural heart disease is a source of extensive research. It's fascinating, and there's a reasonably good mechanistic understanding of the origin of some of these arrhythmias. And as it turns out, some of these arrhythmias can be targeted very effectively with catheter ablation procedures, interventional procedures to cure them. But I always try to put this up, especially when I see the next generation of trainees and fellows. When you look at VTs and serious arrhythmias and acute ischemia, they're all arrhythmias that happen in the absence of structural heart disease, perfectly normal hearts. And it's worth remembering that our knowledge of arrhythmias in that setting still continues to be very weak. So let's deal with structural heart disease, because at least that is an area where we've been able to have some impact clinically. So in structural heart disease, you can see arrhythmias in a variety of conditions. By far, the condition that has been studied the most extensively in clinical practice is post-MI scar-related VTs. But if you look through this list of these conditions, vast majorities of these conditions are substrates that are created by the residual effects of disease processes that leave behind an electrical footprint of the heart. The heart requiring cell-cell conduction for propagation of impulses. Any disease process that sort of interferes with that and leaves behind its footprint sets the stage for future VTs. And some of these arrhythmias, we are now able to get much better understanding of the substrate. And we've been able to actually treat some of these tachycardias with interventional approaches. And we are now at a point where we can actually take some of this knowledge and apply it back and forth with basic science groups. And we think that this is an area where there's going to be a lot more interaction between basic science and clinical EP yet again. I put in a few slides, although Igor had to take a flight to Minneapolis later today, but he could see this in the recorded version of this. I promised him there'd be a few history slides. And of course, our entire field owes a huge debt of gratitude to this gentleman, born to missionaries who lived in Indonesia, interestingly. And he went on, of course, to the world because it's the one diagnostic test that has stood its test of time, and over 100 years, it still is our starting point for virtually everything that we do in clinical electrophysiology. And of course, you come from a center where you've taken the same technology and now you're able to do three-dimensional mapping with it. You know, so ECGs tend to be very powerful. It still gives us. This is 2 slides adapted from John Miller. Both in the presence of structural heart disease and even in the absence of structural heart disease, it gives us an incredibly good starting point. And if you were to fast forward this and apply things like ECG to this, you're now going to have an incredibly good understanding of where the myocardial activation of a given VT starts from. And later, if you can interpret a mechanism, then you can come up with a treatment paradigm on how to fix that problem. So that brings this question of pathophysiology. So again, a quick historical detour. We have a great sense of gratitude when you look at the work of this gentleman who died very young, thought to have died because of self-experimentation in his lab at an incredibly young age. But look at his contributions to the field: re-entry, vulnerable period, alternates. It's just, you know, things that Mines has contributed to the field is quite remarkable. And of course, there was no electrical mapping those days, and for some of the students, you actually should pull out some of these papers and read them. The way research was done was using jellyfishes, and when parts of the jellyfish was cut out and you created preparations like these, mechanical mapping and watching a wavefront propagate was how re-entry was actually studied in those days. So you could crush a part of it, create slow conduction, and really the concepts of macro re-entry were established by some of these experiments. Of course, this is an actual slide from Mines' paper. This is turtle hearts where they have an AV preparation, and this actually shows the entire macro re-entrant circuit in a heart and the way they came up with identifying excitable gaps, if you will, in re-entrant circuits. So that leads to the sort of the 2 major classifications we use in our minds for anatomically defined re-entry, where you actually have an anatomic barrier with a re-entrant circuit that uses parts or regions surrounding that barrier for VT, or you see functional components where effectively without an actual anatomical barrier, you see have a functional barrier that can sustain re-entry. Now, although these are nice cartoons to put up, what always happens in clinical practice, which is the reality, is both these components have a very complex and continuous interplay. In fact, there is not a single macro re-entrant tachycardia, which I'm willing to even take a bet, which does not have a functional component. Almost all arrhythmias have both structural and functional components. And as it turns out, that's the whole paradigm of how you have to envision this. So even atrial flutter, for instance, is one of those macro re-entrant tachycardias that's been so well studied. It's the classic arrhythmia that we use, but it's always maintained by functional components that sort of force the circuit around a given region. So it's worth remembering some of these facts, because with all the new knowledge we're getting, we're going to go back to some of these findings, and we're going to recognize the importance of functional components of arrhythmias more and more as we try to understand substrates and modify substrates. So the knowledge itself in this field, of course, for historical interest, was dramatically improved by the fact that this was a path-breaking paper published in 1968 by Bertel-Heller, where they were able to record a transmembrane action potential non-invasively using voltage-sensitive dyes. And of course, this was subsequently followed by this great paper by Di Salama, where a cardiac action potential was recorded. So experimental ways of investigating the heart gained a lot of attention in the 60s. And another important outcome in this case of a conflict was a group of physicians who were able to work in a public hospital in Staten Island. They were able to, of course, come up with a catheter-based recording of intracardiac activity. So these are sort of the baby steps that gave us clinical electrophysiology. So this is the big citation classic by Tony D'Amato and Ben Sherlock. A next major advancement, it's fair to say, in the last decade and a half has been the power of understanding substrate using electron-atomic mapping in humans. This is data from Frank Marchelinsky's group. And of course, Frank has been a real pioneer in our field to really take many of these complex substrates and start using some of this data for management of VT. And it's important to remember that in the list of structural VTs that we saw, post-MI scar VTs were sort of the first substrate that was effectively tackled with catheter ablation. Substrate, understanding the substrate, mapping the substrate became very important because in clinical practice, it is getting to be somewhat uncommon to see slow VTs with large scars. So we are now seeing patients with more and more hemodynamically unstable VTs with patchy scars, where it is simply not feasible to keep someone in a ventricular tachycardia to map them. So that is becoming a less common type of patient that we're dealing with, and that's where substrate-based mapping and understanding of what it is that we see in the substrate has helped VT mapping. So what about structural components? Here is a heart. I'll show you this twice during my talk. This is a heart of a patient who had a large myocardial infarct, who underwent catheter ablation of VT. And 4 years after catheter ablation of VT, underwent a cardiac transplant. So we actually had a unique opportunity to have a certain set of hearts. We are in a hospital that does about 80 to 90 heart transplants a year. So we had a good collection of some of these hearts. And what you see over here is stretched out thin myocardium. And this, of course, is the rich circuits, areas of slow conduction, things that can sustain VTSC. Now, as it turns out, these types of scars can be well visualized using imaging techniques. And we can now also electrically map them. So here's data. This is a simple MR delayed enhancement image showing you myocardial scar. What you see over here is a punched out region in this frame is a myocardial scar. For some of the postdocs and fellows who don't see this kind of data on a regular basis, this is the whole concept behind it. It sort of deals with sort of differential imaging of a MR contrast that washes in and washes out of myocardium. So if you were to look at areas that are dense scar, the wash in of contrast is slow. The corresponding wash out of the contrast is also slow. Whereas normal myocardium follows the kinetics shown over here, which is labeled here as remote myocardium in squares. It washes in very quickly and washes out very quickly. So this gives you a unique window of opportunity where if you were to time your image acquisition, say at a point when normal myocardium has washed out its contrast, whereas the core of the infarct still has not washed out. So let's say you time an image at this point. The areas where you see contrast still in the myocardium are areas that have slow perfusion. So I'm overlapping this image right here for you. And what you see over here is that this time point, what you see over here is you still have contrast that is sitting around in deep within the scar, whereas normal myocardium is completely washed out, so that's why it looks black in this image. Now, this data has been sort of vastly extrapolated, and there is some reason to suspect or believe that there are some areas that are in between dense scar and normal myocardium, which is the so-called gray zone, and that has received a lot of attention of late in the basic science literature. We did some animal studies, and I'll show you data from that. And as it turns out, if you very carefully look at wash and wash out kinetics, you probably can identify areas that are not dense scar but are clearly not normal myocardium either. So the concept is that perhaps these are regions where there is an interplay of some living myocytes and also scars. So potentially areas that may have harbor circuits for ventricular tachycardia, which would be the implication for us as interventionists and surgeons. So this is the data, and in fact I was quoting this to the fellows earlier today. This is a Schmidt paper, this is from Hopkins, where they use the gray zone analysis. What you see in panel A is a dense scar, which is pseudo-colored as red. And the area that surrounds it, which looks sort of black in this image, when you look at contrast wash and washout kinetics, actually shows the gray zone. And you can see that this gray zone sort of overlies the dense scar, and it's also an epicardial aspect of the ventricle. So that again, highlights another finding in our clinical EP world, and that is if you can actually map the outer surface of the ventricle. So if you do simultaneous epi-endo mapping, you can actually pick out these areas, and that's why epicardial mapping has had a very dramatic impact in increasing the success rate for catheter ablation procedures for ventricular tachycardia. So what is at the CBAC Center in WashU, what are all some of the aspects of knowledge that we've gained? We've learned a lot in the past decade and a half. We've learned that monomorphic ventricular tachycardia circuits are all deep within the scar. The so-called scars, most of the areas of interest for us in the world of catheter ablation is areas less than 0.5 millivolts. It's the area that 15 years ago or 12 years ago we would have excluded from the map. We would have said, oh, all that is dead. No, that is actually the area where you actually find late potentials and sort of the footprints of electrical circuits. Scar border zones are critical because that's where the VT from deep within the scar exits the myocardium. But it's also very important because that is an area where you probably have the greatest myocyte fibroblast interaction. And single PVCs that cause VF, they are rare clinically, but when you see them, they typically come from border zones. So late potentials provide a footprint to the target, and this has been one of the biggest advances in the past decade for catheter ablation of VT, because you can actually ablate the patient in sinus rhythm. You don't have to keep them in ventricular tachycardia to ablate. And of course, with all this knowledge, we are now realizing that patients who have non-ischemic cardiomyopathy electrically have a scar that's very different from patients who have post-infarct cardiomyopathy. In fact, there's a paucity of late potentials. We don't have too many targets to ablate VTs in them. And this really is where I think this whole concept of the interplay between structural and functional components play a big role. And I think functional components will be very important for VT, especially in non-ischemics. So a quick foray through this. This is old data, but it's a classic. It's a good paper for people to read. This is Nabil Elshev, Ben Sherlock data from the 70s. So this is a canine post-infarct model, and what you see over here is the onset of VT, and this is in a post-infarct scar. You see areas of slow conduction and continuous electrical activity, things that initiate tachycardia. Now, if you were to create an animal model, and there's very good data in this, here is data. In this case, we have created an infarct. There are two ways in which we create animal infarcts in our group. One is by microembolization in the LAD territory, and the other is by using an angioplasty balloon and occluding a circumflex coronary artery, doing occlusion reperfusion, something that mimics the human infarct more closely. And what you find again is, even when you go into dense scar, which is shown over here, this is, of course, we have epi and endo data, but even in dense scar fibrosis, you still see surviving islands, small tracts of myocardium. Whereas this is, of course, remote myocardium, you see there's no scar over here. So it's these parts of the deep within the scar circuit that we look for in the world of catheter ablation. And sure enough, when you actually look at this both in animal models and in humans, here is a 3D MR of a apical LAD territory infarct. In this case, it's a porcine infarct. So this is an epi-endo map. And when you look at the epicardial maps, look at what you see over here. This is a surface ECG, and these are the local electrograms that are being recorded from deep within the scar. And look at the timing of these. These are potentials. And here you can see that this area of the myocardium has activated. It's closer to the next QRS complex. although it was driven by the preceding QRS conflict source, slow conduction can be incredibly slow. And in many of these sites, you can also see it's a continuous electrical activity that it takes. The electrical conduction is really discontinuous in that region. And many of these late potentials are areas which would be very critical for a VT circuit. So if you go deep within a scar and face of these late potentials, you may see a long spin to QRS delay as the impulse travels within the scar and exits out at the edge of the implant. So this is the kind of data that we've been using in clinical EP to map and ablate. And just to show you how it's done, this is data that just got published this month. So we take patients who, we map epi and endo. For the past decade, we've been doing epicardial mapping in patients who have not had previous cardiac surgery. Percutaneously, we get into the pericardial space. And some of the research fellows could actually see that in your clinical EP lab. It's now established a standard procedure in America for VT ablations. And in patients who had previous cardiac surgery, our cardiac surgeons open the chest in the EP lab, either an anterior thoracotomy or a subxiphoid surgical incision. And once you get that, this is how the maps are created. So this is a schematic showing you areas of what we consider to be normal myocardium. We use the University of Pennsylvania criteria. Areas greater than 1.5 millivolts are considered to be normal. Areas between 0.5 and 1.5 millivolts are the so-called border zones, abnormal electrograms. And areas below 0.5 are deep within the scar. But even deep within the scar, sometimes you see islands of surviving myocardium. And if you now can map at 1,000, 1,500 points for a given surface of the ventricle, you can now start picking up islands of surviving myocardium, and you can pick up late potentials and scars.
Excuse me, what is your scale resolution? Space resolution?
I'm going to show you that.
Okay.
We have about, it's up to 1.6 to 2 millimeters. Because once you have 1,500 points, you have overlapping sampling. So it's a minimum of 2 millimeters, at least with the catheters that we use. Just a quick word about how we do this. So here's a classic example. You have, this is standard. This is not anything new, but we have another manuscript on methods on this. This is an epicardial catheter. We do double transeptal, like how you do AFib ablations. So this is one transeptal for ablation, another transeptal for mapping. These are multi-electrodes. So you can map both surfaces, endo, epi, and of course, with multiple electrodes, if you can pick out late potentials, and if you have a brief run of VT, you can actually study what happens to late potentials during a VT. So this is a double transeptal approach for mapping both sides. And here's a classic example of the type of data that you see. So this is from what Shiro and Rod did in our group. What we would do is you would have templates of clinical VTs. Sometimes you have 12 leads, or you induce a VT and you get a template of the types of VTs you get in a given case. Many a times you have a cycle length of the VT from the ICD recordings of the patient and get a shot. Now, here is an example of a pace map. And what you do is, you can pretty much, when you map within the scar, you can identify areas of surviving myocardium. And you can also go sub-map within those regions and find out the types of late potentials you see. So in this world of mapping, areas where you see very, very late activations, split activation like what you see over here, or you see an isolated late potential in this case, 220 milliseconds after the local activation is occurred. These are all areas where you tend to see the rich targets for catheter ablation. Now, all VTs have a late potential somewhere, but not all late potentials represent a circuit that can sustain a VT. Does that make sense? So if you were to use an approach where you just ablate late potentials, you're probably going to be overdoing it, because some area is having slow conduction for you to have a macro-oriented VT. But not all late potentials necessarily are representative of areas that are critical for a VT circuit. Now, that is a Gedanken study. That is never going to be done clinically. We could never, there are some patients who have way too many late potentials, so it's very hard for us to sort of attach a physiologic significance for every one of these late potentials. But those are typically the kinds of things that eventually, with more powerful computing and the kind of work you're doing over here, you can actually take clinical data and come up with a priori. You can even live try to understand scar geometry and what is likely to cause VT. It's not going to come from imaging, it's going to come from electrical data. So this is the exact same of that data, except that in this case you see an epicardial surface. You see that? This is an endo map, this is an epi map. You see that the map is, overlaid on this, and again, sure enough, here you have face map, classic VTs, and these are all the areas we target in Ablate. And this is one of those areas where we almost we are a form of a minimally invasive surgical program. The only big problem that we have with epicardial ablation is the presence of coronary arteries. If we are near the phrenic nerve, we place balloons in the pericardium to physically move the nerve away when we ablate. So, as it turns out, many of the surgical tricks are things that you can use in the EP lab for catheter ablation. But the science here is tracking late potentials. Those are the targets. So, what have we learned about late potentials? So, we have learned that, for instance, they typically tend to, this is, collated data from several cases. When you look at deep dense scar versus scar border zones and sort of surviving islands, you typically tend to see very late potentials shown in this part of the graph. They typically tend to be seen, near border zones or surviving islands within a scar. So that's why it's very important. If you see an area less than 0.5 millivolts, you have to map that even more carefully. And what we do in that mapping is we actually set the field thresholds from 50 to 500 microvolts. So some of these signals are extremely small. They are sometimes 50, 60 microvolts, which is 0.05 millivolts. So it's something that if you don't even set the field threshold, you wouldn't even see it. So it's very important to set it in a way that you can actually identify it in sinus rhythm. The same concept actually works even for atrial arrhythmias and especially these complex congenital substrates and so forth. So these tiny little potentials that you see in sinus. Now this is a busy slide, but I'm taking the liberty to show it to this group. This is paper that was published this year in JACC in the month of May. It's a very dense paper. It'll take a couple of hours to read it. I don't want to sort of force anyone to go through that difficult process. But there are some very important findings in this paper. We looked at all patients who came to us with unstable VT, ischemics, and compared them to non-ischemics. And there's a very important finding in this paper, and that is patients who have ischemic cardiomyopathy have more targets, late potentials, ways in which we could target the substrate and sinus rhythm. Whereas patients with non-ischemic cardiomyopathy, it is somewhat disheartening. They have a lower incidence of late potentials. So let me walk you through this particular graph. You can see that this is non-ischemics, ischemics, endocardium, epicardium. So we have two types of late potentials, moderately late and very late potentials. And when you look at the proportion of late potentials, I've told you that very late potentials they are absolute criminals. They have to be taken down. So these are terrorists in the heart. So if you were to look at ischemics and non-ischemics, as a rule, if you just look at epi versus endo, based on earlier data from Reimer and Jennings and others, epicardium tends to be a very interesting area because there's a greater proportion of late potentials that are seen in the epicardium. And again, see how this data is plotted. This is voltage. So this is 1.5 all the way down to 100 microvolts, and this is 50 microvolt bin. If you look at this dotted line, tells you the cutoff of 500 microvolts or 0.5 millivolts. Bulk of the late potentials, almost all the late potentials you see, the entire action is below this thing. Above this point, you can see that there's a paucity of late potentials. So the entire game begins when you have the 0.5 millivolt region. So these are areas that would appear red on your Carto and Avix map, the way you set your thresholds, right? And if you start binning them between voltages, the lower the voltage gets, the more likely you're to see very late potentials. So this is the painful part of mapping, where you actually have to do it much like how you do a patch cam study. It's harder to do, but once you train your lab and everyone to do it, you can do this in 20-25 minutes. You get all the data. And if you were to compare ischemic to non-ischemic, you can now see that in non-ischemics, there is a paucity of late potentials. So you don't have that many targets to take down. And in this particular study, we also looked at outcomes. The success rate for catheter ablation of VT in patients with post-infarct ischemic cardiomyopathy is 82%. I mean, in these patients, it's not like AFib. You have real data. You have device logs. There's good data to follow up. Whereas non-ischemic cardiomyopathy, the success rate in this patient, we had a mean follow-up of 12 plus minus 16 months. Their success rate was 52%. So it was somewhat disheartening. It's still meaningful. It helps. You can still target some VTs. But non-ischemic cardiomyopathy, as a rule, when we see there's no coronary disease, even though they have a very diseased substrate with evidence of scar, a complex substrate, the electrical footprint of whatever disease that caused the scar and the change in the substrate is very different. It leaves behind an electrical substrate that's very different. So this is like an important point to remember, because this is the kind of data in the past decade that has made us think one step further, which is when the substrate is more complex and we don't have that many targets, that's one of the reasons which sort of forced us to start looking at neuromodulation, neuraxial modulation, to go one step above the heart, to really start looking at dynamic factors such as nerve neuromodulation. So that is the nice segue into neuromodulation. And again, this is for Igor Efimov, so that you have to tell him that I did show a lot of history slides. This comes from Mascagni's painting. And for those of you who go to University of Iowa, they actually have one of the few pieces in the world of the actual real version of this slide, this painting. So the structure that really interests us are these white things in this painting, the nerves. And it's now going to be the passion for the rest of our professional careers. So what about neuraxial modulation of VT? So let's spend a few minutes about this. Almost all cardiovascular functions are explicitly modulated by the autonomic nervous system. Chronotropy, inotropy, dromotropy, lusitropy, anything. I mean, it's obviously mammals have evolved this to be able to deal with dramatic changes in heart rate. I mean, when you look at things, right? I mean, all of you have heard in these basic science conferences, you have whales that have a heart rate of 20 to bats that have a resting heart rate of 600 and can increase their heart rate to 1200 per minute during flight. This, by the way, was recorded in the 1960s. So when you just look at mammals and how they can regulate their cardiovascular function, autonomic nervous system is vitally important.
Oh, hold it. What was the last one on the list there? Lusitropy?
Lusitropy. Lusitropy is relaxation of the heart.
Oh, okay.
So, diastolic relaxation of the heart. So, I wanted to sound like a professor. So, what about sympathetic control? There are so many gaps in our knowledge in cardiac autonomic control. Predominantly, I'm going to talk about sympathetic regulation and steps in which how we can modify it, because now we're going beyond catheter ablation, right? So, sympathetic regulation of the heart pretty much occurs due to nerve fibers that come from the neuraxis. There are higher centers, then you have brainstem centers, then you have the spinal cord efferents that go to stellate ganglia on either side. And from the stellate ganglia, you have fibers that actually go to the heart. Now, a third of, a lot of stimulation of the heart pretty much comes also from circulating catechol, which in this case, the entire adrenal medulla is like a huge, it's one large postsynaptic vesicle. More correctly, synaptic vesicle, which when there is sympathetic stimulation, when there's efferent impulses that go to it, it just throws out catecholamines. So both these things play a very important role in the heart. Now, a word about anatomy. This is where, when you discuss with surgeons, cardiac neuroanatomy is incredibly complex. So when you look at published studies, it's mind-numbingly complex, and of course, when you look at data, it is amazing. So you have sympathetic and parasympathetic fibers. As they come close to the heart, they form a plexus. So just to show you how amazing it is from a anatomic perspective, what you see in orange is sympathetic, green is parasympathetic. The purple is a mixture of both sympathetic and parasympathetic fibers. So the heart itself has a huge number of ganglia And of course, the parasympathetic system, there are cardiac reflexes. And the heart is thought to have a huge amount of interneurons. Just to give you the complexity of the system, 60% of the neurotransmitters in the heart are non-adrenergic, non-cholinergic. So our understanding of cardiac, neuroanatomy is
One question about
physiology is
pain.
Sorry?
Pain receptors.
Sorry?
Pain receptors in the heart.
Yeah.
What about do you know something about the distribution of
Cholinergic receptors.
no, pain.
Pain receptors. Well, there is data on that, but it's incompletely understood.
Who is responsible for this?
Which one?
Who is responsible? What kind of nerve system responsible for pain in the heart?
It's the efferents from the heart can be carried through the vagus, nervous system, and some of these plexuses are thought to communicate back to the neuraxis, to the sensory part of the spinal cord, and they all go to the posterior columns and go to the CNS. So many of them are thought to be within blood vessels. So great points. These are all mega areas where our knowledge is very hazy and weak. But like the proverbial guy who lost a key while walking on a long dark road, who ends up searching for his key under a street lamp because that's the only place where there is light. You could have lost your key far along the path. That is the story of cardiac autonomics, but at least I'm glad to say that there are one or two street lights on this road which are still useful. It's very depressing when you think about it. It's so complex that some of the finest minds in basic science stay away from it because there are too many controls. This is too messy. It's very hard to put this in a bilayer. But then you also have a group of investigators in institutions like this who are going and doing human studies again. When you go to Igor's lab and you see Dr. Rodi's lab, it's great to see collaboration with the surgeons. They're actually putting on human hearts and studying it. So this is going to be a very exciting time when we're going to learn about these things. And I'll show you at least one or two street lights where there is something fun we are seeing. So this is again the same data to tell you generally the kind of inputs that come to the heart. So you're right, there's both afferent and efferent. We are predominantly talking about the efferent level here. So what is the big picture? So what should all of you, some of you have engineering backgrounds, some of you of course are from my side of the fence and this, from the medical school side. What should you know about sympathetic nervous system? This is the big picture. The heart is just a pump. Its job is to maintain circulation. It has a very important difference compared to skeletal muscle and the simple fact is anytime there's an injury, an infarct, an inflammatory process, there is a weakening of the heart, which is myocardial dysfunction, heart failure. But unlike other muscles, cell-cell communication is vital for cardiac electrical propagation, and that leads to arrhythmias. But from here to here, you actually have this entire sequence of sympathetic activation, which affects everything from the higher centers in the brain and all the way down to the heart, this neural remodeling. And now, that is the whole link between scar to ventricular arrhythmias. And here's an example where you have a merged CT image. Here you have a scar. So we talked about how deep within the scar you actually have monomorphic VT circuits. Now we know from Stefan Rohr's work and this fascinating work of fibroblast myocyte interactions that scar border zones are also, and this is an actual case that we ablated. They are few and far between, but they're remarkable cases. Here's a single PVC that causes VM, and again they tend to come from border zones. Between these two extremes, you also have functional VTs. So we'll talk about them. So what do we need to know about scars? This is data that Peng Chen and Mike Fishbein gave me, and this has fascinated us a lot, and that is, when you look at infarct border zones, and you look at what happens to nerve traffic in that region, there's nerve sprouting. So nerves tend to grow into infarcts, and there is a greater density of nerve neurotransmitter stains at infarct border zones. Now, we don't know how functional these things are in humans, and that was one of the big efforts of our lab, and we were able to get some data on that. So the same scar, slow conduction, but these border zones now actually have nerve sprouts. So there is a differential innervation of this region. How does that affect arrhythmias? That's where, an entire lifetime of work from Doug Zipes comes into play. So what they have shown is, and of course, when you look at the combined data, not from his group, but many other groups in the world, this is what we know. When you have a scar, that area gets denervated. But even when you have, systemic inflammations, conditions such as hypercholesterolemia, parts of the myocardium get effectively functionally denervated. And anytime you have these kinds of differences, local action potentials, which are dramatically regulated by the sympathetic nervous system, tend to show big differences. So anytime you have significant differences in regional control of action potentials, you set the stage for arrhythmias, especially when you have a trigger. So if you have a myocardial infarct, that is a stage where normally innervated and hyperinnervated regions, or denervated and normally innervated regions, can interplay back and forth, giving rise to sudden death. So this may explain the famous Zipes question of why someone dies on a Thursday morning. We think that you have a fixed substrate, and autonomic regulation can really unmask and amplify some of these problems. So the way we would sort of put together a big picture is this is a simplification, but it's not a crazy oversimplification to say that disease processes can create a substrate, a heterogeneous substrate to begin with, macro, micro level, ion channel changes, structural changes. But there's also this big neural remodeling that we talked about, activation of the renin-angiotensin system. So that causes dynamic modulation. Now, if you look at all the therapies that we use, which have been shown in the past 2 decades to reduce risk of arrhythmias and sudden death, beta blockers, ACE inhibitors, aldosterone antagonists, statins, CRT therapy, bipacing, they all have significant impact in reducing the sympathetic drive on the heart. So this would be sort of one paradigm to look at, sudden cardiac death and ventricular fibrillation. So as we drew in the lab this morning, so you can have QRS, complex related ways of stratifying sudden death. You can look at the ST segment, which is repolarization, P-wave alternance to look at sudden death. Then you can look at beat to beat, one QRS to next QRS. So these are all the ways, electrical ways in which we measure, we are looking for ways to stratify. Again, dynamic modulation, the nervous system. So let's talk about regulation of VT circuits. This is where the fun starts. Now we need to do something about this problem with this knowledge. So we now know that sympathetic tone powerfully influences inducibility of VT, sustainability of VT, degeneration of VT to VF. And we also know that interruption of the autonomic nervous system at many levels tends to reduce VT and VF. So we know this from animal models, and we also have some good human data to support this. So anytime you're dealing with this type of substrate, focal VF, macro reentry, functional reentry, very complex substrate, start thinking of going one level above. Let's go above the heart to start regulating the autonomic input to the heart itself. How did our knowledge evolve in this? This, of course, our Italian gentleman, Dr. Schwartz, over many years did some very fascinating studies in a canine model of infarction. And he actually showed that in these dog models, you can actually, you can recreate what looks very similar to human infarct VF by creating infarct in dogs and then causing coronary occlusion and creating VF. But he also showed something very interesting. That is, when you create these types of infarcts, and this is of course an example of what happens at the end of exercise when you do coronary occlusions, you can create very serious malignant arrhythmias. He also went on to show that in the exact same animals, if you have an infarct but you also do left stellate ganglionectomy, you can see that the same occlusion does not give rise to serious arrhythmias. So here you see the arrhythmias, here you don't. So this is data to show that if you at least reduce the efferent sympathetic nerve traffic, because the stellate ganglion has only sympathetic efferents to the heart. And the only place where you can catch it is near the stellate ganglion, because as it gets closer to the heart, it's this cardiac plexus, and it's anatomically very hard to differentiate these fibers. So We actually, this is where our sort of interaction with our surgeons was very fascinating. Bill Lax, my colleague at UCLA, was doing a lot of transplants, and our transplant group gave us access to the entire transplant database. And this was a this was a very painful study, but it gave us some very interesting findings. We were amazed by the fact that transplanted patients, post-transplant hearts, when they had coronary occlusions, it was very uncommon to see BF. They typically had only asystole or PEA. So we thought we need to quantify that. So this was the study that at that time one of our medical residents, Marmar Musegi, went ahead and did. And we worked closely with Kabashigawa and Conroe and the transplant group. So here's an example of a patient who was on a treadmill post-transplant, was exercising on a treadmill and suddenly developed PA and collapsed. And this was the rhythm that was recorded. So this is a dramatic case. The patient was, CPR was started. We paid the surgeons and said, oh, you need to put in, ECMO, do whatever it takes. And the guy was dying, okay? So he was taken with ongoing CPR to the cath lab. And the very first coronary injection is shown over here. You can see that, This, of course, for all the cardiology fellows in the audience, this makes you vagal looking at it. So this is, our surgeons are not that phased by this. Whatever it is, they handle it. But this is left main occlusion. So this is what traditional teaching tells you, that if you occlude the coronary, you should be in VF. But this heart was not in VF. So we went ahead and quantified our result. So we looked at a whole lot of patients. In this case, we looked at 628 transplants out of 1,500 transplants that we had in our registry. But we had very high quality data on 628 patients. And we classify these as sudden and non-sudden deaths. So when you looked at the sudden deaths, when you looked at these patients, you found that VF was very uncommon. The commonest rhythms that were seen were asystole and PEA. This tells you that, my God, nerves probably have something to do with the ability to fibrillate. We've seen very similar results with atrial fibrillation too. We've never seen sustained AFib in a transplanted heart. If we see AFib, it's almost always cardiac rejection, ongoing inflammation. So fibrillation is very rare in a transplanted heart. And this is just quantifying this data showing VF was rare. And in fact, we took patients like the case I showed you before, where we actually had, acute ischemia, which was documented. This is people who died in front of us in a cath lab. And again, when you look at these cases, you see asystole and PEA, where the most common rhythm in VF was very uncommon in a transplanted heart. So that's background data for you. But now how do you try to modulate this? So the way we modulate this is to try to go to a higher level. So the first level of modulation happens in someone with BT storm. You try to sedate them, intubate them, keep them in ICU, and you almost induce general anesthesia. So that is a way of controlling the higher centers. So our way of joking about this is to make the mind not matter. It's not mind over matter, it's to make the mind not matter. That could help. Vast majority of the time, that controls VT. Now, what about, and it's not uncommon that when you look at any protocol for electrical storm, you reprogram device, you look for things that you can fix, put in a balloon pump, you do things like beta blockers, sedation, and inducing general anesthesia. These are important steps. And we'll talk a little bit more about thoracic epidural and stellate removal. Again, things that we learned from the surgical world. So, now let's come one step below the CNS, higher centers. How do you do spinal modulation? So spinal modulation can be effectively achieved by thoracic spinal stimulation. And so this is stimulation of these efferent nerves, which are pre-ganglionic, right? These are fibers that are pre-ganglionic to the stellate ganglia. So what happens is, there is very good data for this, stimulating these nerves. And in fact, this is classic data, again, comes from Doug Zipes' lab. They actually did a small randomized study in canines with healed MI. When they did spinal cord stimulation of the T1 to T2 segments of the spine, and then subjected these animals to 2-minute ischemia. And what they found in their study, and of course, when you look through the entire paradigm, it was pretty remarkable that when they subjected these animals with spinal cord stimulation, they found a significant difference, much like the Schwartz study, on incidence of BTBF. So it tells you that spinal cord stimulation actually had a very positive effect on arrhythmia incidence. Now, you can also achieve the same thing by modulating something within the spinal cord itself. So you could actually use thoracic epidural anesthesia, or intrathecally you could use pharmacologic agents such as clonidine. So both these things can also achieve the same thing. And of course, this is data that comes from also in Zipes' group where they use intrathecal clonidine. And this is again in an animal model. So this has not been done in humans, but we did something very close to this, and that is to use thoracic epidural anesthesia in patients who came in with VTBF stop. So we had some patients who were so unstable that we thought they would die in being transported from the ICU to the EP lab. So now there is some data to say that if you do any kind of spinal modulation, we don't quite realize what could be the mechanism of benefit. It could be complex, it could be multifactorial changes in myocardial perfusion, maybe even a change in parasympathetic tone. Because we always focus just on decreasing sympathetic tone. But does it work? So this was the index case, and this was a patient in 2005 who was airlifted to us, and we thought this patient would die before we could even get this patient transferred to an EP lab. Just to show you the kind of arrhythmias, this is the patient sitting in the ICU. This is the time when cardiologists call 911. They call EP, right? So this patient was dying, and we plotted just to look at the shocks versus time. And this patient's ICD battery died out after a while. An ICD gives you only a limited number of shocks, 100 to 120 at most, depending on the device. And these were all external shocks, and this was escalating. And at this point, just out of sheer desperation, the patient was intubated as an IV amnio, was anesthetized, and out of sheer desperation, we tried thoracic epidural. The patient was already anticoagulated, so we could not inject just lidocaine into the percutaneous vein with the left stellate ganglion, and that actually had a dramatic effect. So this really encouraged us to start using thoracic epidural more consistently and sort of earlier on in our protocol for VTBF stop. And here's the very same patient. What you see over here is the epidural catheter. And in this case, we've injected contrast through the catheter to show you T1, T2. So you can use local anesthetics, bupivacaine, or you can use opioids. It doesn't matter. Either one of those medications in the epidural space can control arrhythmias. And if you look at cardiothoracic services, many patients are walking around on their floors, telemetry floors post-cardiac surgery with thoracic epidural catheters. Do you use that stuff in your program for pain control post-cardiac surgery?
We've used some of it.
Some of
it's
it.
a bit of an issue with the heparinization. We have used it in some of the minimally invasive cases.
Our anesthesia guys allow us to use it after the catheter is placed. So they've never instituted when someone is anticoagulated, but after they place it, they don't mind if you anticoagulate. So, in fact, that was one small advantage of thoracic epidural. If they place the catheter, later you can anticoagulate. Whereas if you do the percutaneous stellate ganglion block, if someone is anticoagulated, it's very hard to go re-inject when someone is anticoagulated. So, interestingly, this, Doug Zipes at that time wrote an editorial about this, and this became a Class 2B indication for neuromodulation. But then we continued this kind of work, and we said, why not try to also look at ganglionic modulation? There is already good data on ganglionic modulation. We talked about Peter Schwartz's work, but we also had data for ganglionic modulation from an earlier paper. This came from Nadimani's group, where they looked at patients who were in VTBF storm. And it was a complex study. They looked at ACLS-based management of VTBF storm versus using sympathetic modulation using beta blockers. And in this particular study, I'll just focus on the key point of their study. What they showed was anytime you use beta blockers or you use left stellate ganglion blockade, in this case it was percutaneous, they were able to have a big effect, and this is of course shocks. And you can see that these are of course the frequent flyers, in 80 shocks we had per day. These were people in extremes. And of course you can see that left stellate ganglion block reduces shocks. And of course in this case you can see when it dissipates, the shocks come back and you can reinstitute block, it continues. So there was clearly a sort of a cause and effect, if you will. So this is data to show that ganglionic blockade works. And of course, in their study, they also showed that IV beta blockers are more effective than antiarrhythmic drugs. Here's an example where you look at VTVF episodes versus intervention. So beta blockers, sympathetic ganglionectomy, these things work. So that led to this study by us. And this is I would give a $5 gift to any of the fellows who can name this volcano, this unpronounceable volcano which screwed up air traffic and all of Europe last year. So VF stop. So this is the paper that we recently published in our group. We looked at our combined experience of using thoracic epidural anesthesia and surgical left stellate ganglionectomy. And these were patients who were incredibly sick. These are patients you can see, had multiple VT therapies, failed EF, some of them with very bad hearts. And this pretty much gives you, the number of therapies. You can see this particular patient, sarcoid cardiomyopathy, had 200 therapies. These were extremely sick patients. These are people we thought, death was imminent. And sure enough, and these are also patients who had, complex. Some of them had monomorphic VTs, multiple monomorphic VTs. Some of them also had polymorphic VT. So, but interestingly, a lot of them had monomorphic VTs of multiple morphologies, showing multiple circuits. Not all classic polymorphic, because we always think polymorphic sympathetic modulation. This was a sort of a learning point for us. And this is the way the procedure is done. The way our surgeons do it is videoscopically. You can do it supra-spinal, or you can go between the ribs with independent lung ventilation, and they sort of take down one lung, and you can cut out the sympathetic ganglia. Of course, it doesn't, it's very hard sometimes. Fat, everything looks the same. When it's actually removed, they do a frozen section in the lab in the OR, before the patient leaves the OR. And of course, this region you have to really carefully dissect out the sympathetic chain. Now, this is the data, the combined data, thoracic epidural. This is data of thoracic epidural in all these patients. This is therapies before TEA, this is therapies after, during thoracic epidural anesthesia. When this left stellate ganglion was removed, this is the data of the effect of left sympathetic denervation. And what you see over here is, irrespective of the type of arrhythmia, whether it's polymorphic or monomorphic, there was a 50 to 60% reduction in arrhythmias after stellate ganglionectomy. There is a small window post stellate ganglionectomy when there is aggravation of arrhythmias, but pretty much the net effect is a significant control of arrhythmias. And these are some of the most complex substrates. In the past, we were using this only for long QT syndromes and patients with polymorphic CPVT, but this is now using it for more complex substrates. So, parasympathetic modulation, a word about it, it's incredibly important. It deserves a whole day of lecture on its own, and it's a story that's still emerging and one that we don't understand that well. But it's worth remembering that some of these, interventions is not as pure as we think they are. So, I'll conclude with one or two slides just to show you. This is the next two slides show like 3 years of work from our human lab. We did a very extensive set of human studies, which was very difficult to do and perhaps will never be repeated again. But it had to be obtained, and that is to look at what happens to sympathetic regulation of ARIs as a marker of action potential duration in humans, both in normal, in cardiomyopathic patients, and we also use normal controls. We reflexively stimulated the nervous system by infusing nitrite. So these are patients to begin with who are dying of VTs and due for VT ablation. So you had to stimulate the sympathetic nervous system by giving them nitrite to, dump their pressure. But it's actually a very powerful way to stimulate the sympathetic nervous system. And we actually looked at what happened with nitrite and what happens with isoproterenol, which is direct sympathetic stimulation. So one big finding from the study is when you look at any region of a heart, remote myocardium, border zone, or scar, all these areas showed very poor response to reflexivity stimulation. So even an area that had voltage above 1.5 millivolts showed denervation. So cardiomyopathy actually causes ischemic cardiomyopathic hearts tend to have global denervation, not just the scar that's denervated. And in fact, the more quantitative data from this particular study showed that when you look at isoproterenol stimulations, these areas were also areas where there was significant evidence of denervation supersensitivity. So when you look at non-treated area, and I'm not going to belabor this particular point, the sum total of this is it shows that these are areas of the heart. So what we think as normal myocardium voltage-wise are areas that show significant autonomic dysfunction. And in our study, what we have found is, this is a 50-page manuscript, which actually shows circulating catecholamines may be quite deleterious in patients with ischemic cardiomyopathy, because it's not just the denervated zone and the border zone. So the remote myocardium also shows significant denervation supersensitivity. So just a quick flyby, the types of works we are now doing both in organ experiments, which we discussed one-on-one, and the data that you'll be seeing from our group is we now do microdialysis in whole organ experiments, where this is a microdialysis catheter, so we actually circulate and we collect neurotransmitters with microdialysis. So we've now standardized this. We've done this in porcine experiments where we stimulate the stellate ganglia. And it's pretty much when you step on the gas pedal, you try to find out how much neurotransmitter is released. And we're trying to understand how the right and left stellate ganglia, how they control myocardial sympathetic tone. So there are some patients who have benefited by having a right stellate ganglion removed after left stellate ganglionectomy, because thoracic epidural is slightly more effective than just left stellate removal. So that makes us think that if you go one level above, you're able to control both sympathetic ganglia. So this is an area where there's a good area to do mechanistic research and collaboration with surgery, because it's almost like leaving a temporary pacing wire when patients come out of cardiac surgery. You can actually do microdialysis and measure neurotransmitter levels in the human heart regionally. Typically people measure this in the coronary sinus, but this you can actually measure at the myocardium itself. And I'll just skip by this data and say that the next translational step for this particular study and where we're taking this data is the PREVENT-BT trial, which we've designed, and we're in the process of trying to get funding, industry and